By studying the virus and understanding the genetic structure of the virus, we'll be able to know if the virus is changing, if the virus is mutating. We should be able to identify the clusters of human-to-human transmission, and then that will be very helpful in terms of helping public health interventions. So I think, today epidemiology is no longer conventional epidemiology. Today epidemiology is more like genomic epidemiology, because genomic epidemiology takes beyond the realms of conventional epidemiology by telling you exactly the areas where transmission is occurring, telling you exactly how the virus is evolving, and then that becomes very important in terms of the in terms of diagnostics, in terms of vaccines if you do have, or in terms of therapy if you do have, because we all know that these are very fast and highly mutating pathogens. So the question is, can you deploy vaccines, drugs, and then diagnosis without understanding, how fast or how fast these viruses are changing and the areas where the viruses are changing. If the viruses are changing in areas that are being targeted by your primers for your diagnostic, then you end up having a lot of false negative. If the viruses are changing in the areas where your drug is targeting, for instance, let's assume there is a drug for coronavirus, then it means that that drug is not going to be effective. If the viruses is changing in areas where your vaccine is targeting the epitopes of the virus or the antigens, then it means that your vaccine is not going to be working because there will be changes, and then the epitopes are not going to be recognized. So I think that becomes very relevant holistically when it comes to control of this outbreak. So I think we'll work together with the Nigeria Center for Disease Control and other partners so that we can all do this together for the benefit of Nigeria. So the question is, when you have in the same environment several imported infections, there's a possibility that if you have cross transmission, that you can have recombination, right? So that we have to be very mindful. So if you have viruses coming from various places and then they're all in the same environment, as a biologist, I understand that there's a very possibility of recombination if they are all within the same people or within the same environment, and then we end up having a completely different virus. That's very possible. One of the centers that are designated by the Nigerian government for diagnostics, we've been testing people or suspected cases from Osun State. We have some from Ekiti State, so we've been testing. Our turnaround time is probably about 3 to 4 hours because we have a lot of machines, and we also have a lot of manpower. I think the best tool that can help you to answer that question is actually looking at sequencing, because if you sequence, if it's the viruses there, it will tell you how the whole nature of the virus is, and then the reason why probably it was inconclusive. Or if it is a contamination in some lab, then the sequence will not find anything and will find other pathogens. So at least that's clear evidence that there was nothing in that particular sample.